Hi, I am Jose Blanca. This is an introduction to population genetics, and today we are going to talk about Hardy-Weinberg. There are certain ideal populations in which we have a constraint that allow us to calculate the genotypic frequencies from the allelic frequencies. Remember that in the general case, if we have the genotypic frequencies, we can calculate always the allelic frequencies because it's very easy to count how many alleles there's in each genotype. But in the general case, when we have the allelic frequencies, we cannot infer the genotypic frequencies, okay? But there are certain ideal populations in which we can do that. Also, in these populations, there will be no evolution. That means that there will be no change in the allelic frequencies. Which are the characteristics of these ideal populations? Well, they have to have random sexual reproduction, that it's called technically panmixia. So that the mating is not related to the genotype, the degree of the relationship between the individuals of the geographic locality, so it is really at random. And also there are no microevolutive forces going on that we will study which are these microevolutive forces, but mainly is that there will be no drift. That means that the population has to be infinite, has to be or at least a very, very large number of individuals, that ideally will be infinite number of individuals. There will be no selection going on, no mutations, and no migration. If you have all these constraints, then there's a relationship that allows you to calculate the genotypic frequencies from the allelic frequencies. Imagine that we have a population in which we have three possible genotypes: capital A, capital A, lower a, lower a. These are the frequencies of the population in any at any point of time. Okay, we start with these genotypic frequencies. Then you can calculate how many gametes will be generated in the population, because you have that the capital A individuals will generate only capital A gametes, lowercase AA individuals will generate only lower A gametes, and of course the heterozygous individuals will generate half and half. So the frequency of the A gamete is exactly the same as the frequency of the allelic frequency, that means P, and the frequency of the lower A gamete will be Q. Okay, now we can do we can calculate how many genotypes will be generated in the next generation from these gametes, and the result is that in the next generation you will have that the frequency of capital A is P squared, the frequency of lower A is Q squared, and the frequency of capital A lower A is 2PQ. These frequencies, these genotypic frequencies, depend only on the allelic frequencies of the previous population, of the previous generation. They do not depend on anything else. So given some allelic frequencies, you can calculate the genotypic frequencies. This happens only in these ideal populations in which you have panmixia, no drift, no selection, no mutation, no migration. Okay, this result was the first milestone in population genetics. I was discovered by Hardy and Weinberg. So these frequencies, these proportions of genetic frequencies are called Hardy-Weinberg proportions, and they are very, very, very important in population genetics. So in a population that follows the Hardy-Weinberg proportions, you will have that the frequency of capital A is p squared, the frequency of lower a is q squared, and the frequency of the heterozygous is 2pq. Real populations do not need to follow these ideal frequencies. It is true that in animals, usually the real genotypic frequencies are close, usually quite close to the ideal Hardy-Weinberg frequencies. But that is not the general case. For instance, if you have a population in which your individuals reproduce by a strict selfing, then the genotypic frequencies will be P and Q for the homozygous individuals and 0 for the heterozygous. Okay, so it is not for all populations that you have the Hardy-Weinberg frequencies, only for the case in which you have an infinite population with no migration, no mutation, no selection. Okay, only in that case, and random mating, panmixia. Only in that case you will have this. But it is true that animal populations tend to be more or less like that. One thing that you can do, I have added an exercise in which you have the frequencies of 3 genotypes. This is a classic paper published in 1971. It's one of the first papers in which we had genotypic frequencies. So you can go there, and from the genotypic frequencies you can try to calculate the allelic frequencies and then the expected genotypic frequencies if these populations were in Hardy-Weinberg, and then you can check if the difference is big or small. Here you have the solution to this exercise. Also, if you assume that your population is more or less close to the Hardy-Weinberg genotypic frequencies, you can use that information in order to infer genotypic frequencies that you cannot measure. For instance, if you have a disease in which you can see people that is homozygous for the deleterious allele, so people with the disease will be homozygous for an allele that it's a diseased allele. For instance, in this case, we have frequencies for Friedreich's ataxia, that it's a recessive disease, and you have in different European populations how many individuals do have the disease. With that, you have the frequency only of the recessive allele. Of the genotype with homozygous for the recessive allele. But from there, if you assume Hardy-Weinberg, you can calculate the allelic frequency and all the other frequencies. Okay, and this is what you have in this exercise. So if you want, you can do the exercise, and there is the solution. In this case, we are not checking that we have the Hardy-Weinberg frequencies, but we are assuming that the population is more or less following these Hardy-Weinberg frequencies, and we can use that assumption to estimate for instance, how many heterozygous people it's in the population. one conclusion of Hardy-Weinberg is that when you have alleles in low frequencies, for instance, in this part here we are representing P and Q, okay, in the x-axis, and in the y-axis the frequency, the allelic frequency, sorry, the genotypic frequencies. So when you are close to fixation, and remember that most loci will be close to fixations in most populations, then you can see that the allele that is in the lowest, the minor allele, will be mainly in genotypes that are heterozygous. So you will have very few homozygous alleles, homozygous genotypes. This is a conclusion that you can derive if you assume Hardy-Weinberg. And I remind you that Hardy-Weinberg, for animals at least, it's quite a good assumption. This has some implications, for instance, in diseases. Usually, the alleles that cause a disease will be in low frequencies. So most people carrying the disease allele will be healthy heterozygous if the disease is recessive. So when you have a recessive allele, even if it's deleterious or even if it's advantageous, it won't matter for selection, because most of the time it will be invisible for selection. But because if the allele is recessive, it will be, according to Hardy-Weinberg, mostly in heterozygous individuals, so the allele will be almost invisible for selection. Okay, this is another consequence of Hardy-Weinberg. As you see, this is a very easy to get theoretical result, but it has deep implications for real populations. Also, you will hear about the Hardy-Weinberg equilibrium. That is related to how allelic frequencies change with time, so how populations evolve. Let's see. Let's assume that we have a population in a generation 0 in which we have the allelic frequencies, so the genotypic frequencies in the next in the next generation will be the frequencies set by Hardy-Weinberg. But what happens in the next population? Will be these frequencies changed? We can calculate the allelic frequency in the next population from the allelic frequency in the previous from the genotypic frequencies in the previous population, and you can see that the allelic frequencies in the next generation will be exactly the same as the allelic frequencies in the previous population, previous generation. So the thing is that if the population follows the Hardy-Weinberg restrictions, it's an ideal population that follows these restrictions, the allelic frequencies won't change with time. So there will be no evolution. The population will be completely stable. This is called Hardy-Weinberg equilibrium. Okay? Neither the allelic frequencies nor the genotypic frequencies will change with time. Even when there is no panmixture, if there is no mutation, no selection, and no migration, and no drift, there will be no evolution. This is a more general result, but so if there is panmixture, you have the Hardy-Weinberg equilibrium. If there is no panmixture, but you have the other microevolutive forces not acting, there will also be no evolution. Okay, now let's see what happens when there's some deviation from these assumptions. In real populations, we won't have all the assumptions completely met. So there will be some deviations from the Hardy-Weinberg proportions and the Hardy-Weinberg equilibrium. One of them is that real populations are not infinite in the number of individuals, okay? That is something that it's quite clear, and that causes something that it's drift. Also, you will have selection, you will have mutation. All that affects to the genotypic frequencies. But let's see a couple of exceptions that are not related with the microevolutive forces, but because we will talk about the microevolutive forces next week. Let's think about what happens if in a population we have endogamy. So we don't have panmixia. That means that the reproduction is not completely random. For instance, in the case of plants, you have endogamy if you have self-implants. Or also you can have population substructure. So you can have a population that is divided in, for instance, two valleys. And the individuals from one valley and the individuals from the other valley, they do not make completely at random, because the individuals in one valley tend to breed more with the individuals in that same valley. What happens then? What happens when there's population substructure is that there's a lack in heterozygous individuals, and that is called the Walhun effect. So imagine that you are analyzing a population that has some diversity, but although you don't know it, this population has substructure. You have two subpopulations. Imagine that we are studying a locus with two alleles, and in order to make it very easy, let's imagine that these two populations do not mix at all. These will be two completely isolated populations. Now, we are unaware of this, and we study the genotypic frequencies of these populations, and we see that in one population we have only homozygous individuals for the capital A, capital A genotype, and in the other only homozygous individuals for the lower A individual. When you add all up to calculate the genotypic frequencies for the overall population, what you see is that you have 0.5 genotypic frequency for capital A, and 0.5 for lower A, and no heterozygous. So there's a clear lack of the heterozygous individuals when you compare it with what you expect according to Hardy-Weinberg, because in Hardy-Weinberg what we would expect in this situation is that we had 0.5 heterozygous the frequency for the heterozygous genotype should be 0.5 according to Hardy-Weinberg, but we have 0. So there's a clear lack of heterozygosity. This is what will happen when you have substructure in your population. There will be a lack of heterozygosity. There's different ways of studying this lack of heterozygosity, and Wright, a classical population geneticist, developed some indexes that reflect this lack of heterozygosity in different circumstances. For instance, what happens when there's subpopulations or when there's inbreeding. For the case of subpopulations, we can use FST. Okay, FST relates the heterozygosity expected for the whole population and the mean heterozygosity found in each population. And this is the definition of FST. you remember that we talk about FST when talking about the genetic distances between populations. Okay, but this was the first approach to FST, an approach that still can be used. Also, there's a Hardy-Weinberg deviation when you have selfing. If you have selfing, imagine that you start with a population that has only heterozygous individuals. If you self that population, if you reproduce each individual with itself, okay, with time you will have less and less and less heterozygous individuals, and at the end you will have a complete lack of heterozygosity. There will be 0 heterozygous individuals, and all individuals will be homozygous. This will happen with time, okay? So if we have a population with complete selfing, eventually there will be no heterozygosity at all. So again, we have a lack of heterozygosity when we compare with the Hardy-Weinberg expected genotypic frequencies. This is one of the cases in which Hardy-Weinberg is useful. It's not just useful to estimate genotypic frequencies from allelic frequencies when you assume Hardy-Weinberg. It is even more useful when you have genotypic frequencies and you compare your real genotypic frequencies with your expected genotypic frequencies. So endogamy as a substructure has a similar effect. It reduces the number of heterozygous individuals. It reduces heterozygosity. Any inbreeding will reduce heterozygosity. And another statistic is related with this effect. If you assume that your reduction in heterozygosity is due to inbreeding, you can calculate how many expected individuals you would expect in an outbred population, that means in a population that follows Hardy-Weinberg, and you can compare that with the heterozygosity that you have in your population. And using that, you can calculate a new F that in this case is reflecting inbreeding. It's the same formula as before, the formula for FST, but the formula for FST was applied when you assume that the reduction in heterozygosity was due to substructure of the population. But in this case, we are assuming that the reduction in heterozygosity is due to inbreeding. You can think about this fixation index as if you had two contributions to the genotypic frequencies. One due to individuals that have crossed, and another one that is due to individuals that do not cross at all. Okay, this would be an extreme population in which you have two kinds of individuals. One that are in Hardy-Weinberg, so they behave in panmixia, mating randomly, and another part of individuals that would be the F part of the population with a frequency of F that completely self, so they are completely inbred. And these are the genotypic frequencies that you would expect in a population with this degree of inbreeding, if you call F inbreeding. We also talk, when talking about diversity, remember that we said that the expected heterozygosity was a very good measure of diversity, of genetic diversity. The expected heterozygosity is the level of heterozygosity that one has in a population that follows Hardy-Weinberg. There's Other deviations, for instance, if you have selection, for instance, if you have a disease that selects against the homozygosity or the minor allele that it's deleterious, you will have a lack of some homozygosity, the homozygotes for the disease allele. This is an example in a paper in which the authors check the lack of homozygosity in one gene. They did that for several genes, and they found that in several genes in the human genomes, there's 0 homozygous in the Iceland population. You can calculate how many homozygous individuals you will expect according to the allelic frequency, and you see there's none in the population. That indicates this lack of heterozygosity indicates that maybe there's something going on with these mutations, okay, with these alleles. These alleles do have less homozygous individuals as expected, so maybe they are selected against. In fact, in the case of this gene, they found some homozygous individuals outside the Icelandic population, and they had problems. They found severe intellectual disability and other problems in these individuals. So yes, the prediction of Hardy-Weinberg is not met because this is really problematic. And also they found that in other genes that in which they had a lack of homozygosity for the minor allele, they found that this allele causes miscarriages. So a very serious problem because the individual is not even born. And that's it for the Hardy-Weinberg equilibrium. We will continue next week with genetic drift.